Okay, recording begins, and it is July 23rd. All right, so here we are, folks, comfortably seated in week 3 of the 5-week semester. We're on Wednesday right here, and today we are going to begin and finish our discussion on photosynthesis. 31 slides, we got an hour 50, shouldn't be too hard to get through, okay? Any extra time at the end of today's lecture, I will open up to study guide questions. All right, so we've got questions about the lecture midterm, or I guess even the lab midterm, but I recommend we focus on the lecture midterm, you know, because there's a lot of stuff on that. It's 33 questions. It's not a short exam, really. Today, we've got your lab midterm, your lab quiz, as I call it right here. The lab quiz, as I mentioned before, it's going to be open notebook. It'll start at 1, and I have a hard out at 3:50. I got to get to an appointment at 4. So if you take the full 3 hours at the end, I'll be like, hey, I got to get out of here 5 minutes. This doesn't matter. It won't happen to any of you. You're going to be fine. You're going to all do great. Any questions in general about where we are in the course? Cool. Tomorrow will be our lecture midterm exam. All right. Don't forget to get a Scantron 882E and bring a pencil. All right. I'll have some backup Scantrons, but I might not have enough pencils for everybody. Next week is just a straight up regular lecture week. Okay? There's no exams, there's no midterms, anything like that. We just talk cell cycle, and then we talk meiosis and sexual reproduction. This is when we start using the Stuff of Life book, which I have as assigned reading for summer school. I'll still have the corresponding Campbell biology recordings posted for you if you prefer to listen to those. If you got a copy of the Stuff of Life, you're sitting pretty. You know, you can just read that thing. I also have a PDF version of it posted, but it's a manual scan PDF, so it's kind of crummy. But there's no ebook version of this thing that I could try to, like, rip for you or something. Plus, I don't do that because I don't want to get in trouble. Just buy the Stuff of Life. It's freaking great. It's really incredible. You still have time to get it and order it. It's like the first half of our 340 genetics course in a comic book. It's wonderful. Check it out. I promise you, you'll be happy you have it if you continue on into genetics. And then in our final week of class, we'll have our lab final and our final exam, along with, you know, wrapping up a discussion of cell cycle, meiosis, and then natural selection and inheritance and genetics. Really, next week's cell cycle and the week after is just heavy genetics to prep you for the next bio course you take. Any questions about that? About our what do they call it? Our roadmap for the rest of the semester. Okay. Then in that case, let's just go ahead and open up our PowerPoint. Yes, enable external content. Okay. So it's Wednesday today, right? Yeah. Yesterday, on Tuesday, we talked a ton about respiration. Before that, we talked purely about energetics. And then we jumped into enzymes. And then we jumped into specifically respiration, which, remember, carried out in the mitochondria. We describe it as a series of redox reactions. Redox reactions involve electron transfers. Here I am. I'm holding an electron. I transfer the electron onto something. That reduces the thing that got the electron, and I got oxidized because I lost an electron. Okay? Cool so far? I'm just gonna imagine that we are cool so far and just keep talking unless you have a question. Okay? In this process, I serve as the reducing agent because I reduced something. The benchtop serves as the oxidizing agent because it oxidized me by taking that electron off. Fair? Okay. You can think about electron transfers as transfers of energy. When an electron goes from one thing to another thing, all right, it gives it the capacity to have more order, more structure. It therefore has more energy. Also, electrons can exist at different energy states. All right, they can be high energy electrons or low energy electrons. And if an electron goes from high to low energy, well, that represents a loss of energy, and that energy has to go somewhere. Even if it's just radiated as heat or light, like we saw in our plant blood demo in the back of the room. All right. Cool. Oxidation, reduction, electron transfer, redox reactions. Where does it all occur? In the mitochondria, across the inner membrane. All right, 3 stages. Glycolysis, stage 1. Pyruvate oxidation plus a citric acid cycle, stage 2. Oxidative phosphorylation, stage 3. Oxidative phosphorylation includes the electron transport chain and chemiosmosis. The electron transport chain pumps the protons, generating the proton motive force. Chemiosmosis harvests the proton motive force using ATP synthase, all right, to generate ATP. You get a little bit of ATP from each step here. Following stage 2, the majority of the energy is in high energy electron carriers, NADH and FADH2. I just want you to know the net outputs of stage 1 and stage 2, really, because the real processes are so much more complicated than this. I don't want you to waste your time memorizing an oversimplified model. Okay? Cool. Here's one of those big money slides, I call them, right? Where it's got a lot of information, and if you understand it, it indicates you understand the key steps of the process. Stage 1, glycolysis, a little bit of ATP, substrate level phosphorylation. Stage 2, complete the breakdown of glucose, generating CO2, all right? And a little bit of ATP via substrate level phosphorylation. The majority of the energy following stage 2 is right here in these electron carriers. That energy gets pumped into the electron transport chain, driving chemiosmosis in the process of oxidative phosphorylation. Oxygen is there at the end of the electron transport chain as the terminal electron acceptor. It can be the terminal electron acceptor pulling electrons down the electron transport chain because it is so electronegative. When it accepts those electrons, it becomes water, and that's like the output. Now the electrons are at low energy. You've harvested as much of the energy out of the electrons as you can. Cool. Yeah, this is where the majority of ATP comes from. There we are. Boom, boom, boom. It's all right there. Boom, boom, boom. Electron transport chain, pumps protons, chemiosmosis. This is also called the proton motive force, the separation of concentrations of protons. Remember, any system where if you put disorder or concentrate something, it wants to fall back to equilibrium, and that's a source of energy as well. If you push the molecules that are trying to reach equilibrium, or the atoms that are trying to reach equilibrium, through something, you can harness energy, like a dam. All right? Cool. ETC. That's funny, because it's like saying et cetera, but it's the electron transport chain. That's fun. That just happened. Yeah.
The NAD plus and the FAD that
Let's get them.
off.
Right here?
Yeah.
So it's
The electron transport.
the first thing is, this is just a bad placement of a plus symbol. This is NADH.
Not right there.
You mean...
After,
I'll find you. I'll find it.
at the electron, yeah. When they come off, do they go back and circle into that cycle again?
Essentially, there's an enzyme called NADP. There's an enzyme called NAD plus reductase that's responsible for reducing NAD plus to make NADH. And there's an enzyme called FAD plus reductase, right? And so these molecules get reacted with by an enzyme to generate more of the reduced electron carrier. Right now they're in their oxidized form as NAD plus and FAD plus. Does that sort of make sense? Okay, cool. But you don't have to really know their fate for exam purposes. But they go into the mitochondrial matrix and they get used again. Sick. Okay, chemiosmosis, there it is, spins the rotor, ATP. Same slide we've seen a lot, and that brings us to fermentation. And here we are with new material. Okay, so if there's no oxygen available, then the electron transport chain cannot operate normally. It needs oxygen to continue pulling things through the electron transport chain. If oxygen is not present, the electron transport chain backs up. If the electron transport chain backs up, there's no way to generate the proton motive force. The proton motive force is quickly harvested by ATP synthase, generating ATP, but then you're out of luck. You know, you've got all your ATP that you were going to get, and the proton motive force is depleted because there was no oxygen. This is why you don't want to be without oxygen for too long, okay? But things can survive under anaerobic conditions. Some things can survive pretty indefinitely under anaerobic conditions through a process called fermentation. Now, you carry out fermentation in your muscle cells. If you're working out a bunch and you burn all the ATP in your muscles, you carry out a specific type of fermentation to keep generating ATP. But fermentation always generates an undesirable byproduct, okay? It always produces an undesirable byproduct, which then your body has to deal with. You know about lactic acid buildup inside of your muscles, right? It makes your muscles ache. I know it's not really what happens, but don't worry about it, okay? And then we've also got alcohol fermentation, okay? And alcohol fermentation is primarily carried out by yeasts and bacteria. Okay, cool. These get their names from the end product of the process of fermentation. So here's what's going on, okay? I've told you before how you get a little bit of ATP, okay, from from glycolysis. You get a little bit of ATP from substrate level phosphorylation. Fermentation is sort of a last-ditch effort to try to get more ATP, all right? Here's the problem. In order to split glucose into pyruvate, right, you actually need NAD+, all right? This can get a little tricky to think of, so bear with me. This NAD+ is going to pull electrons off of glucose and generate NADH. And usually, that's awesome, because NADH then goes through the passes electrons through the electron transport chain, pumping protons, and you have no shortage of NAD+. But if there's no oxygen, you actually run out of NAD+, and all you have is NADH. So fermentation exists really to regenerate NAD+, because if you have NAD+, you can keep driving glycolysis and getting a little bit of ATP. But what do you do with the electrons on NADH in order to generate NAD+? Well, you put them onto those pyruvate molecules, okay? Now, these pyruvate molecules, in normal cellular respiration, if oxygen's available, they've got a bunch of energy in them that you're going to harvest, right? By driving it through the Krebs cycle, okay, you get a bunch of energy out of that, all right? But when you reduce pyruvate with NADH, all right, you transfer the electrons onto pyruvate, reducing it, you generate this useless byproduct. In this case, it's lactate in lactic acid fermentation, and in this case, it's ethanol in alcohol fermentation. So You get 2 out of a yield of 32 ATP molecules from this, but it's better than nothing, and you do it out of desperation to survive, okay? So this is great, right? You can just run this process in a circle, but the problem is, right? The problem is that now you got to do something with this molecule. And there's a whole system of enzymes in your body that have to do with dealing with lactate, all right? You also flush it out of your system. That's why people be like, you got to keep exercising, keep your blood flowing to move the lactate out. It's an oversimplification, but it's fine. Let's look over here at the example of alcohol fermentation. So in alcohol fermentation, it's a little bit different. Different enzymes do it, right? You reduce pyruvate, and you actually kick off a couple of carbon dioxides, all right, generating these ethanol molecules. But both of them had the same net result of giving you NAD+, which is essential to continue splitting glucose and generating ATP. Does that make sense? Both of these are technically like toxic, all right? So they're bad things to have. So you took something that was going to give you a bunch of energy, you turned it into poison, basically. All right, cool. This right here is why, I don't know if people still do home brew, but in home brew, they've got the little bubbler, the little water trap at the top. Air bubbles out of it. If you don't have one of those, the fermentation tank explodes. All right, that's because of the carbon dioxide released here during the reduction of pyruvate into ethanol. That's fermentation. How do we feel about it? It's all right? Cool. So that's all the fermentation stuff I wanted to talk to you about. I've mentioned this before, but we always oversimplify our discussions of photosynthesis and cellular respiration by talking in terms of glucose, all right? And glucose is great. Glucose runs all the way through the process of cellular respiration, through every step, okay? But other molecules can be broken down into chunks that can feed in later into the process. So remember how fats were a glycerol molecule attached to 3 fatty acid chains, right? The triglycerides. Well, that glycerol molecule, if you split a fat up, that glycerol molecule can be converted pretty easily into an intermediate molecule within the process of glycolysis. So you don't get as much energy as glucose, but you get some energy from that. And then those fatty acid chains, they can be broken down and fed into the stage of the precursor, the molecule that goes into the citric acid cycle. Okay? So you don't need to know the specifics of where these things go, all right? But you should know that you don't have to only break down glucose in order to carry out cellular respiration. You've probably known somebody on a low-carb diet or a no-carb diet, and they don't just lose the capacity for producing ATP. The body breaks down other things like fats, and then out of desperation, like proteins. All right? Cool? This is, basically why you want to eat a lot of protein when you're on a low-carb diet. Yes?
Does that also explain why it takes longer to digest the different macromolecules, like how carbs are kind of seen as like a long-term energy source because they have to go through every cycle versus proteins being a quicker energy source? They only have to go through 2 or not necessarily?
Not necessarily. It has more to do with these arrows here of which enzymes are carrying out the breakdown of the fats and proteins and how are they regulated, right? Because they're all being regulated via inhibitors and activators, right? And your body has a slow time of adding or removing these activators and inhibitors, right? So it's more about this step for that. Yeah, your body regulates to initially go for the carbohydrates because they're the highest source of the energy. But it's not really any of this that has to do with the speed at which they're broken down. It's just your body doesn't break them down. Is that cool? Answers? Awesome. Okay, here we go. This is the big summary figure. You ready for it? I like these. I try to include them a lot. Cool. Cellular respiration generates ATP, which is energy for cellular work, by a process called chemiosmosis, which uses a proton gradient. That's sort of our already, this bioprocess doesn't really work there. That's connected there. Sorry. Let's not I'm going to get too ahead of myself. Let's just go through it, and I'll have you engage with the slide. Here we go. Cellular respiration has 3 stages. A is glycolysis. B is the citric acid cycle. You could also say pyruvate oxidation and the citric acid cycle if you wanted to be really specific. You could also say the Krebs cycle, and that'd be fine. All right? And then C, this one's tricky, oxidative phosphorylation, by a process which produces many ATP, by a process called chemiosmosis, which uses a proton gradient. Okay? Protons diffuse through ATP synthase, and that's in the in oxidative phosphorylation. Okay? Cool. It's a little bit of a circle there. Let's jump over here. Cellular respiration oxidizes glucose and organic fuels to 2 molecules here. Yeah, carbon dioxide. Water and carbon dioxide. If you ever forget, just think about doing this. And like what comes out? You know it's CO2, and you can feel the disgusting water. It's all from inside your body. It's gross. It's gross. I try to keep the water that's inside my body, but if you've noticed, it doesn't always work that way. Yeah. All right. Cellular respiration, so we've got this. It's CO2 and it's water. It uses blank to pull electrons down blank. So it uses what to pull electrons down? I heard somebody say electronegative. That's good. What do we know that's very electronegative? Uses oxygen to pull electrons down the electron transport chain, right? Which pumps protons to create the proton gradient. Protons diffuse through ATP synthase and oxidative phosphorylation. Cool? Awesome. Awesome. Anybody need me to keep this slide up at all, or are we good with it? Sick. Now we're finally into photosynthesis. Like I said, it's 31 slides. I think we've got time. I'm not too concerned. Learning objectives, great. You can always check this out. Hey, there's another cycle here. This one's called the Calvin cycle. Whoa. All right. Good. Oh, and check this out. NADPH, which is frustrating. NADPH, it does the exact same thing as NADH, but it has a phosphate group. All right? So just remember P for plant, or P for photosynthesis. NADPH is the electron carrier in photosynthesis. I'll show it to you again, but I just wanted to hit that right here. Okay. Now, as I've mentioned a few times, right, within a photoautotrophic organism, there are both chloroplasts and mitochondria. So there can be this whole little cycle going on within a single photosynthetic organism, okay? This is meant to indicate the ecosystem, talking about how, oh, there's energy coming in, and then you get ATP, and this is what the ecosystem does too, and it's all due to chloroplasts and mitochondria. But it's all about systems, okay? And where we set that boundary up. I know I've hit that point a few times, but I would love it if my Bio 105 grads went out there and they knew what we were talking about when they said, oh, the system falls to equilibrium. It just means we've constrained what we're looking at, okay? So in this model here, we are looking at the ecosystem, okay? And this figure is claiming, just one second, this figure is claiming that chloroplasts in the ecosystem generate organic molecules and oxygen, which are consumed by mitochondria and produce CO2 and water, which are consumed by chloroplasts, produces ATP, and then heat pumps out of the ecosystem, okay, due to inefficiencies in the reactions, in the energy transformations. Question?
You said it's all about systems. When you said that analogy, what do you mean by setting?
Totally. If I can talk about an organelle as a system, I can talk about a cell as a system, I can talk about an organism as a system. But if I'm describing that as my system, then I'm not concerned with the things outside of it. Okay? Yeah, awesome. Now, you might describe inputs into a system and outputs from a system. But you'll notice that this model of the ecosystem doesn't include the chemical reactions that generate the light energy or what the heat does. See what I mean? Solid. So photosynthesis, it directly or indirectly nourishes almost the entire living world. Of course, there are locations on the planet where there's no light, all right? Some of those places receive energy inputs from decaying matter that rains down like into the deep ocean, but there are systems where they aren't reliant on photosynthesis. They're reliant on other sources of energy. That's really more of an aside, but when you see this almost the entire living world, it's fine to think, well, what do you mean almost? Why isn't it the entire living world? Okay. Right here, we have the equation for cellular respiration. Up here is the equation for photosynthesis. If you look at the two, they are the same equation with the arrow flipped. Solid? Cool. So, in photosynthesis, carbon dioxide comes in, water comes in, energy comes in. You break them down, you mix them up, you slap them all together, you reorganize them with that energy, and you go from some little molecules of carbon dioxide and water to a big molecule of glucose, and you produce some oxygen. All right? Cool. Cool. All right? So this is an anabolic reaction. It therefore requires energy. It's also an endergonic reaction because it requires energy. All right? It's going to have a positive delta G value, right? Because the energy of the products is higher than the energy of the reactants. There's our energetics piece right there. And this whole process is going to be facilitated by many, many enzymes. And it's going to have a lot of steps in that little arrow there. Questions on this slide? Cool. Now I do want you to look real close here at this equation is in stoichiometric balance, indicating that the carbon that goes into the glucose and the hydrogen that goes into the glucose and some of the oxygen that goes into the glucose actually comes from these molecules. All right? You're going to actually be putting in enough energy here to break that water molecule. And that takes a lot of energy because oxygen's very electronegative and it's not very reactive. All right? You can slap a water balloon around all day. You can hit it with you can try to light it on fire and the water won't just start burning because water isn't very reactive, primarily because oxygen holds onto that electron so tightly. It's not going to participate in electron transfers freely. See what I'm trying to say? I'm trying to draw it to make everything connect. Am I failing? Am I succeeding? I get paid either way. It's the great thing about this job. Okay. There's a great short story. It's a classic literature short story called The Cobbler in Spite of Himself, where a cobbler is mistaken for a doctor and loves it because if you do a cobbling job wrong, the shoe is bad and you get blamed. But if you're a bad doctor, you just blame the patient. And that's exactly how it is with teaching. I can just say that you didn't pay attention You know, isn't that great? I would never do that to you, though, class. Photosynthesis occurs in chloroplasts in plant cells. The structure of the chloroplast, it's very, very similar to the structure of the mitochondrion. I really try to take my time talking about respiration because the same thing happens in the chloroplasts. You've got a super folded membrane to increase surface area with a bunch of enzymes embedded in it that carry out steps for generating ATP using ATP synthase by harvesting a proton motive force. It's the same thing that goes on in these thylakoid membranes. All right? The only difference is instead of the initial energy source being a high energy organic molecule that you have to split up and break up, you just directly energize the electrons with solar energy. All right? It's pretty cool. It's pretty fascinating. Okay, chloroplast structure. You've got an extra membrane on this one, okay? Which, if you think about it, makes sense if you're considering the free-living origin of this thing, okay? But we can have that discussion at another time if you'd like to. Outer membrane, inner membrane, intermembrane space. But nothing's really getting pumped across the inner membrane here, okay? At least to drive ATP synthesis. Instead, all of that proton pumping and ATP synthesis occurs across the innermost membrane. And that innermost membrane is called the thylakoid membrane. It is stacked into these pancake stacks that are called grana in the plural, singular being granum. All right, I don't test you on Christe or grana or granum or any of that stuff. You'll learn it if you follow cell bio. If you continue through cell bio, you'll get tested on all this stuff. Okay, here's the fun thing, right? If you have more membranes, you have more chambers, and every chamber needs a name. So the outer membrane and the inner membrane, there's an intermembrane space between the two, just like in the mitochondria. But then there is a space between the thylakoid membrane and the inner membrane. And that is this thick, syrupy goo called the stroma. All right? So the stroma is the chamber and the fluid in which the thylakoid membrane is suspended. And then you have to have a name for inside of the thylakoid membrane. Thankfully, gracefully, they call it the thylakoid space. Okay? It's a space inside the thylakoid membrane. I do usually like to throw in a question at you on the exam that requires you to know stroma and thylakoid space and also the direction of proton movement in photosynthesis. If you remember, in the mitochondria, protons got pumped out across the inner membrane into the intermembrane space. So pay attention to where the protons go in photosynthesis as well. And I will highlight it for you. I'll highlight it for you in my lecture. Chloroplast is the organelle. Chlorophyll is the pigment. We already did a little lab on pigments, right? That's pretty cool. We isolated them out of spinach powder, hit them with ultraviolet light, made them get energized by absorbing that radiation, and then they kicked off the radiation as lower energy light. Looked like the plant extract turned red. Cool? Here's a chlorophyll pigment, okay? Check out the structure of this chlorophyll. Now, if you've reached the point where you've accepted that everything's just hydrophobic or hydrophilic interactions or osmosis or diffusion or one of the 4 things that I've highlighted for you, and you looked at this molecule, you might notice, oh, there's a big old hydrocarbon tail on this thing. I know that's going to be hydrophobic. What do I know? Oh, I think there were glycolipids. And glycolipids, they had a big hydrocarbon tail in order to be embedded in the phospholipid bilayer, right? Right? Into the hydrophobic domain. So this hydrocarbon tail here is also used for anchoring. It anchors the chlorophyll into this special protein complex, all right, that has hydrophobic domains. Now, this porphyrin ring structure, okay, you'll see them, if you go through biochem, you're gonna see these a lot. They regularly store energy or do other things. They do a lot of stuff, okay? But in this case, what they do is they absorb radiation and the electrons get jazzed up, all right? So there's this magnesium atom here in the center, which is really key for that jazzing up of an electron, but is that's more detail than we need to worry about for Bio 105. All right, here's a chlorophyll molecule. It absorbs light and it vibrates. Okay, you're going to have a bunch of these all near one another embedded in this protein matrix. They're all going to get blasted with light, vibrate, and then the protein complex is going to focus that light, that energy, that vibration energy, all right, into a single point. It's basically just like a big parabolic satellite, a parabolic dish. You know those, you see the solar things that they have going now, where it's like it's a big satellite dish looking thing, and all the light gets focused up into a top point, and then that point, it usually has some liquid in it that it just boils, and that churns a little spinning thing that generates energy. You seen those? Those exist, all right? It's a pretty good analogy for this, where all the chlorophyll molecules get a little bit of energy, and it all gets focused into a specific point. It gives you enough energy to eventually yank an electron off of a water molecule. Okay. I always have to talk about stomata briefly. They're incredibly important, and some people don't have to take Botany 105 who have to take Biology 105. This is really important stuff, right? We breathe, okay? We have a diaphragm, we make a vacuum inside our body, and it draws air into our bodies, right? And then we pump the air out, we go back and forth, and we get the oxygen we need, and we get rid of the carbon dioxide we don't need. Plants don't have a respiratory system, okay? So they have to rely entirely on diffusion, all right? Diffusion of gases. So essentially what a plant does is it just opens these little holes on the surfaces of its leaves, all right? And it lets gases diffuse in, all right? Now, you're going to be running out of CO2 because this process consumes CO2. So the inside of the leaf will have lower CO2 levels and higher oxygen levels because photosynthesis is making oxygen. And when you open a stoma, I think it's a stoma in the singular, I'm pretty sure, CO2 diffuses in and oxygen diffuses out. But it's all diffusion, okay? Cool? So that rate of diffusion is going to be dictated by the strength of the concentration gradient, something we've been talking about with diffusion. If there's very little CO2 inside and a lot of CO2 outside, the plant can get the CO2 faster, okay? Now this is all fine and dandy, right? But why do they have to regulate it, right? Why does the plant have to have these guard cells that open and close to regulate stomatal opening? Why do they have to regulate stomatal opening with these guard cells? Yes?
There's times when the pressure gradient is like an advantage.
That's interesting, yeah. You want to try to concentrate the CO2 or something? Concentrate it, yeah. Okay. That's really good thinking, absolutely. But I'm looking for something else. Even though it's not necessarily wrong, I am looking for a different answer. So in my mind, it feels like it's wrong. You know, isn't that interesting how that works? When you expect a different answer, no matter what somebody says, it feels wrong. Whoa. Smooth, no big deal. All right. So you open a big hole. Let's say I don't have lungs anymore, and I'm like, well, I need oxygen, so I just open a hole on my side. What else is going to happen? Bad things can get in. That's one. Yeah. Did you both say the same thing? Yeah. Bad things could get in. I don't want a big hole on my side. I don't want it there. Okay? In fact, a lot of pathogenic organisms, especially it's a fungal strategy, what they'll do is they can detect the stomata and they'll just push a little hyphae up and just push through it, wait for it to open and just force their way into the cell and then parasitize the whole plant. So you don't want to always have the holes open. That's one reason to regulate stomatal opening and closure. Question?
Could it also like help with their structure?
It does help with their structure. Elaborate on that.
I was just thinking about like turbidity when we were talking about a few weeks ago in lab and how like things could swell or shrivel up, and I feel like if you're not getting the right amount of what you need to make the system work, it could like mess with the system.
I'm going to give you 5 points, 5 John points for that. All right. So with the added point that is turgidity. Turbidity is how like much is suspended in a solution. We'll nip that in the bud right now. You know, sick. Everybody gets those mixed up. Here's the thing. If I open that big hole on my side, a lot of water is going to diffuse out. And I'm going to be losing a ton of water. Believe it or not, there's more moisture inside of a leaf than there is in the surrounding atmosphere. So yeah, carbon dioxide goes in, oxygen comes out, but also water starts flooding out. And you need that water in order to maintain turgor pressure and in order to maintain the structure of these tissues. So stomatal opening and closure is tightly regulated, and the BIOL 105 explanation is to limit water loss. I better throw one more piece at you here. The time of day when you can carry out the most photosynthesis and you need the most CO2 is the hottest time of the day, when you're going to lose the most water if you open the stomata. All right? So you don't want those things to just open during the day and close during the night. It's not good enough. You know what I mean? Any questions about that? Cool. Here's a cell. It's called a mesophyll cell. There are palisade and spongiform mesophyll cells. They're in the leaf. The mesophyll is the middle part of the leaf. You don't have to know leaf anatomy for this course. Chloroplasts are concentrated in mesophyll cells in leaves. Sick? All right. Hey, guess what, folks? Photosynthesis is a redox process too. Great, right? Let's talk about electron transfers. In photosynthesis, water is oxidized, carbon dioxide is reduced. You can kind of intuit it by looking at this equation of, okay, the carbon in glucose had to come from carbon dioxide. So in order to make that more complicated molecule, it's going to need some more energy. It's going to need some more electrons, right? So carbon dioxide is going to get reduced. It's a little less intuitive to be like, oh, water must be oxidized. But if you think about it like, well, where do the hydrogens come from? Okay? Think about it like that. It's like, oh, they come from the water. It's not entirely accurate, but it's okay for getting the rules down. So water gets oxidized, electrons get pulled off of it, and this generates oxygen. It loses the hydrogens. CO2 gets reduced, right, where it gets hit with electrons, and then it can make a bigger molecule. Solid? So carbon dioxide is reduced, water is oxidized. Is the process exergonic or endergonic? Endergonic, because it requires energy. Energy goes to ender the process. All right? Cool. Here it is in the same representation as the redox. We had a redox figure about respiration that was drawn this way. Here's the same one. Okay, but now it's for photosynthesis. Cool. The energy for the endergonic process, it's provided by light. All right, sick. Oh, sick. I was bummed out. I was like, I don't get to talk about light as much as I usually do. But I forgot that I'd moved it into this PowerPoint. So we're going to talk about light. Now, we're also going to take our 5-minute break a little early because I drank a lot of water. Okay, folks, it's time. We're back, and we are going to talk about light. The light source, so right here, right? Photosynthesis is an endergonic process. The energy is provided by light. I talked to you a little bit about light, but only as an aside. So let's talk about light, what it is, how we discuss its energy. It'll just make all this make a little more sense, okay? So up here, we've got the electromagnetic spectrum of energy, okay? Energy is described as waves, though sometimes energy behaves as a particle. It just indicates to you that neither particles nor waves are a perfect system for describing energy all the time, but it's good enough to let us make predictions, and if it's good enough to let us make predictions, then we continue to use it in the sciences. That's just a tale as old as time. Keep your eyes open for it, you'll see it constantly. Gibbs free energy, that just simplifies describing energy in general into a number so that we can quantify it and, make predictions with it. And it works, so we use it. Every description of everything does not have to be a perfect description. It just needs to be good enough for us to make predictions in science. The energy of radiation is described in wavelengths, right? The distance between a peak and I always forget. The wavelength is the difference between one peak and the next peak in a wave. So a low energy wave has long wavelengths, and a high energy wave has short wavelengths. Cool? We're cool with all that stuff? All right. Let's start over here with radio waves. Radio wave wavelength is meters long, right? It's kind of wild to think about that, the distance between peaks on these. They're not very harmful. They're bouncing around us all the time. They don't pass through our bodies super effectively, and if they do pass through our bodies, they're not high enough energy to interfere with anything. Okay, they don't destabilize anything. Microwaves, special. They resonate really effectively with water. Okay, that's how microwaves work. They use energy in this range to make the water molecules vaporize. All right, you basically steam food from the inside out. That's why bread gets so nasty when you microwave it, okay? Because you both dry it out and you steam it. And I don't know, a steamed bun always looks good when they serve it to you, and then I don't like them. I don't like soggy bread. Okay, then you've got infrared. That's heat. You're all radiating infrared radiation right now, all right? And it's not hurting the people next to you. It's harmless, right? Infrared radiation. But you can visualize it with, say, an infrared camera, special electronics can let you see outside of the visible spectrum. Then we get into, and remember, we're going from long to short wavelengths, low to high energy. Then we get into the visible light spectrum, Roy G. Biv, it's right there, okay? Ranging from 750 to 380 nanometers, okay? Red light is the lowest energy light. If you're trying to remember this, which it's kind of fun to remember, with red being the lowest energy light, it's right above infrared. Infrared means below red, and this is low energy. Above violet is ultraviolet, and ultraviolet radiation can be dangerous. It's higher energy, that's why. Okay? So within this visible spectrum, okay, we're going to be talking about what gets absorbed by the chloroplasts. Okay? Moving up past violet, you've got ultraviolet, X-rays, gamma rays. They get high enough energy all right, and they can pass through more things, and they can provide enough energy when the radiation hits something to cause it to react in ways that we don't like ultraviolet radiation, say making your DNA molecule go click or like chunk, and that's bad, all right? And then you get a melanoma. Okay, solid. Any questions here about radiation and energy in general? Let's move on. Light passes through the leaf, okay? Some light is absorbed, some light is reflected, some light is transmitted, okay? The absorbed light will be the wavelengths that you don't see when you look at the leaf, all right? So chloroplasts, they're not very good at, well, they're not designed to absorb the green portion of the spectrum. So green light can reflect away, bless you, and green light can pass through. Fair enough? And you've already seen the absorbance spectrum of the different photosynthetic pigments because you isolated them and graphed them last week, right? So this is all basic to you. Here's absorbance spectra right here. This is from an old group, like back in 2021 or something, okay, that did the experiment that you did yesterday, just a different representation. That's not to even be from like 2019, honestly. And then here's textbook representations of the absorbance spectra and the action spectrum. Now, the absorbance spectra, it shows you what wavelength of light being on the x-axis, each pigment is good at absorbing, absorbance being on the y-axis. Cool? This one's all good? Then we've got the action spectrum. This tells us which wavelengths of light are best at driving photosynthesis, okay? The photosynthetic activity is measured as oxygen release because that reaction right here shows that this oxygen being released is in stoichiometric balance. So all the oxygen being released can be related to how much glucose is being generated, if you remember your chemistry stuff. How do you generate data like this? Well, you take like an algae, you put it in a chamber, you have an oxygen sensor, you hit it with a certain wavelength of light, and then you see how much oxygen it makes. Am I speaking especially quietly, or is my volume okay for people at the back? I feel like my voice is starting to fail me at this point in the semester. Yes?
Seeing this just reminded me of the figure 1 that we have to do for the pigment lab, and when the follow-along video, you just selected the first set of points, and that's kind of what I did also. I was wondering if that's okay, or if we should do like our own set for our lab bench. I couldn't tell which ones.
No problem. It was A was bench 1, B was bench 2, C was bench 3. That was the, how it's all laid out. But if you already submitted for bench 1's data, bench 1's data was good data, Nice. Your data is so darn good that other benches want to take it. That's pretty cool. Now, as long as you can show me that you can graph it and create the figure and then present it to me, I'm totally okay with it. Yeah. Cool. Yeah, great. Any questions about this stuff? Okay, now we're into the real deal. I've got 55 minutes to talk just photosynthesis with you. Here we go. Brace yourselves, because this is going to get as heavy as our respiration stuff. All right? Cool? Here is, I keep calling it the money figure, and it's starting to feel uncomfortable, But here's the money figure, by which I mean it's got everything I want you to know about photosynthesis over-summarized. So you should be able to look at this at the end of our discussion and be like, oh, I understand what the light reactions are and how they generate ATP and NADPH and how those go into the Calvin cycle and how that generates sugar from carbon dioxide, and then why that releases NADP+, then how the light goes in here and makes the water molecule split, and you get the oxygen out of the light reactions. Photosynthesis is 2 sets of reactions. We will focus aggressively on the light reactions, all right? This is where the light energy gets harvested and converted into chemical energy in the form of ATP and NADPH. All right? Excuse me. Now, there's another set of reactions called the Calvin cycle. When I was a student, they used to call them the dark reactions still because they're not fed with light energy, but they also don't occur in the dark because you need the light reactions to be happening to drive the Calvin cycle. So instead, now we call it the Calvin cycle, named after the primary discoverer, or the person who got named after, whose name was Melvin Calvin, which is, you know, you have to be a scientist if you're named Melvin Calvin, because you're not going to play football, man. All right. The Calvin cycle takes these high energy molecules, ATP and NADPH, and generates sugar. Cool? So we're going to focus here, see how these outputs get generated, and then we're going to hand wave Calvin cycle as, yeah, that energy goes in and it it hits CO2 a bunch and it makes glucose. Okay? Cool. NADH versus NADPH. They're very similar. NADPH has a phosphate group. I'm gonna try to trick you. I'm gonna try to get you. Make you miss a question on the exam with NADH and NADPH. All right? So you gotta know that they're different. You gotta know that one is used in photosynthesis and one is used in respiration. And the one that's used in photosynthesis has got a P right there in the name. NADPH, P for plant, P for photosynthesis. All right? Solid? Okay. Does the same thing. This is the important part of it. All right? Does the same thing. Cool? A biochemist would holler at me right now and be like, and I'd say, silence, biochemist. I'm in charge now. All right. Was that a question? The P is this phosphate group hanging out down here. Yeah. And so it is going to make the molecule do different stuff and behave slightly differently, and different enzymes can work with it, you know. But, yeah. That's the bio breakdown. Photosystems. All right? I really want to frame all this for you. The light reactions are just an electron transport chain and chemiosmosis. That's what the light reactions are. Just like what we saw in respiration. Okay? The only difference, the key difference between them is that the electrons for the electron transport chain in respiration, they started as high energy electrons, right? Because they were in glucose. They were already high energy electrons. All you have to do is yoink them off, all right? In photosynthesis, the electron gets elevated to a high energy state by blasting this protein complex with light. But otherwise, it's the same thing, okay? An electron goes from a low to a high energy state, and then it falls down to lower and lower and lower energy states. That energy is harnessed to pump protons, generating a proton motive force, which is harvested by ATP synthase to generate ATP. The photosystems, there are two of them, are these complexes that are responsible for absorbing light and kicking up a high energy electron. Okay? So I'm the photosystem right now. I'm full of chlorophyll. I'm getting blasted with light, and it's all getting focused into my core. And I've got this electron here, and then it goes, whoa, to a high energy state. And then it goes, boop, 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 boop. And as it boops, protons go shoop and move up through the electron transport chain. And then there's just another photosystem over here that's basically the same as that one. And it's getting blasted with light, and it's got this electron going, and then it goes, bloop, It elevates that electron to a high energy state again. All right? And then that electron just gets put on NADP plus so you can get some NADPH. All right? So it's a little different there, but the electron transport chain here is still the same. This is called the Z scheme because the electron goes to high energy and then step by step to a low energy and then up to high energy. And that looks like a Z, okay? It's the Z scheme for linear electron flow. Now, I intentionally tried to run through that without the accompanying slides because I'm really trying to connect the similarities with respiration. Question, please.
Does it also bind with oxygen when it gets to that low state?
Thank you so much. You can have another 3 John points. That's great. So at the end of it, here's another key difference, right? There are 2 outputs of the light reactions. Those 2 outputs are ATP and NADPH. There's no water, okay? Because after that electron goes broop to high energy and then to a low energy, it gets elevated again to a high energy state, and then that high energy electron gets put on to NADP plus to generate NADPH. So there's not really like a terminal electron acceptor in the same way. All right? The electron falls to a low energy state, but then it gets elevated to a high energy state again. Great question. I'm hoping that this preamble will make the actual figures more approachable.
So is this
Please.
an enzyme in the membrane, or is this a protein?
This is a protein with a bunch of pigments embedded in it. Yep, so this is a big protein with a ton of chlorophyll in it and other pigments. And you can rest assured there's an enzyme in there, here or there. But for our purposes, it's a big blob of proteins with a bunch of chlorophylls, all designed to take all the energy absorbed by these chlorophylls and focus it into this single point, all right, where a high energy electron can get kicked off. Cool. So here's a photosystem. A photosystem consists of light harvesting complexes and a reaction center complex. Light harvesting complexes, all right, are like the dishes of the satellite, sort of, the dish of the satellite, sort of. And then the reaction center complex is involved entirely in kicking up a high energy electron. Okay? All right, questions?
There's already a question about water, but how does the like that higher energy electron get captured? Is it relevant to like pulling apart the H2O molecule?
It's so freaking relevant. You can have 3 points. All right? So this special pair of chlorophyll A molecules, it's going to lose an electron. And then this thing here, it loves electrons. It loves electrons even more than water. All right? So this is going to, with the help of an enzyme, pull the electron off of water to replenish this. It's kind of weird to think about, but imagine that I'm taking a metal beam, you know, my big muscles are swelling up and I'm bending this thing, right? It's a very stable thing, this big metal beam. And if I keep putting enough energy into it and then it breaks, it can really like blast shrapnel, right? So taking the electron that is very, very stable and blasting it up to a high energy state can actually work really well, all right, to get a super high energy electron. It's the analogy that I use, and it's not a perfect one, but it lets me flex and look strong.
That oxygen interaction happens out in the stroma, not like in free space, not within that protein.
Yes, like here. Yep, just like this. Yeah, and it's catalyzed by an enzyme, of course, because every reaction is sort of catalyzed by an enzyme here. And it's more complicated than this, of course. All right, cool. So let's go back to the beginning. here's the photosystem, okay? What does it do? It focuses light energy down into this special pair of chlorophyll molecules in the reaction center complex, kicks up a high energy electron, okay? Then that electron can fall down an electron transport chain to pump protons. Photosystem 2 was discovered second, so it's named photosystem 2, but it functions first in linear electron flow, which is the ideal type of electron flow here. Yes?
That whole system that we were just looking at is photosystem 1.
This here is a generalized photosystem, so it would be the same as photosystem 1 or photosystem 2. Does that make sense? It's just meant to be generic. This is a generic photosystem that just shows you, hey, it absorbs light, kicks up a high energy electron. That's what both photosystems do. So they really look the same. The structure of the photosystems is incredibly similar. The only difference is they're better at absorbing certain wavelengths of light. Yeah. But they both exist to take a low energy electron and kick it up to a high energy state using light energy. Maybe let's see if it helps when we look at the step-by-step. So we're going to do a step-by-step look at the process, and then we're going to look at the whole thing in one big figure, and then we can double back on the step-by-step if there's specific steps that are giving people struggles. Okay, here we go. Photosystem 2. The best, highest energy yielding type of electron flow is what we call linear electron flow. And that's when the electron goes from low to high to low to high and ends up on NADPH. Okay? There's another type of electron flow that we'll talk about, but this is sort of our ideal basic electron flow. Okay, so photosystem 2 is the first one that functions. It absorbs light. This special reaction center, this special pair of chlorophyll A molecules within the reaction center gets an electron kicked off of it, because all of these chlorophylls are absorbing energy, and all the energy is getting focused into this point. Okay? When that electron gets kicked off, this gets a positive charge. It's a very strong oxidizing agent. And as an oxidizing agent, it can yank the electron off of water. Cool? This is why I've been so intent on mentioning oxidizing agents and reducing agents and things like that. So why is this called P680? Because it's the best at absorbing light at the wavelength of 680 nanometers. It's a special pair of chlorophyll molecules that exists to have electrons kicked off of it and then to oxidize water. Okay? Solid so far? So now what do we have? We have a high energy electron. Okay? Then that high energy electron goes down an electron transport chain, pumping protons. And now it's a low energy electron again. And it gets passed on to photosystem 1. Photosystem 1 has its own special pair of chlorophyll molecules called P700 because it's the best at absorbing wavelength of light P700, 700 nanometers, right? Now, P700 is going to get blasted with light to kick electrons off, but it doesn't take an electron off of water. It accepts the electron from the electron transport chain and then kicks it back up to a high energy state. And then it gets transferred to NADP plus to make NADPH. This is a pitfall I don't want anybody to fall into, so check this out. This point here says electron transport chain. It is technically an electron transport chain because an electron gets transported through a couple of intermediate molecules. But but it's not an electron transport chain that pumps protons. All of the protons get pumped as the electron moves from photosystem 2 to photosystem 1. Photosystem 1 elevates an electron to a high energy state so that you can get NADPH. Remember, the light reactions need to generate two things, ATP and NADPH. All right? So the NADPH comes from photosystem 1 right here. The electron goes up to high energy, and then it just gets popped onto NADP+. All right? Cool. We're going to look at the full picture, sort of, yeah. I overdo it with how many different ways, how many different forms of explanation I have for the light reactions, I think. But bear with me. I'm hoping that one of these will resonate with you. There are 8 steps. Of course, there's more to it, but you define how you break your steps up. Photon hits a pigment in a light harvesting complex of photosystem 2. That energy eventually excites P680. P680 kicks off an excited electron. Now it is P680 plus because it has lost an electron. Okay? Water is split by enzymes, and the electrons are transferred to P680 plus, making it P680 again. This generates the oxygen that plants generate when they're photosynthesizing. Now, when this splits, technically the protons get released into the thylakoid space, and that's good because, oh no, it's a catastrophe. But don't worry, I don't think anybody noticed. Sorry for putting you on blast. The protons are released into the thylakoid space. Now, this is a highly evolved system, and this is good because these protons are going to actually contribute a little bit to the proton motive force. So remember near the beginning of our discussion, I said, I will expect you to know where the protons get pumped into. This right here should be enough of an indicator that the protons are all going to get pumped into the thylakoid space. All right. Oxygen gets released as a byproduct, and it can't be held because oxygen is a nonpolar small molecule which will basically freely diffuse across the phospholipid bilayer. So it gets released. Next, the electron transport chain. You don't have to memorize the electron transport chain pieces until you take plant physiology, where you have to memorize like all 15 components of this whole process, which is rough. All right. But I believe in you. You can do it. Electrons fall down an electron transport chain, lower energy state. This is just respiration style, right? They're pumping protons. There's an ATP synthase. Cool? It's just everything we talked about with respiration. Chemiosmosis. The electrons end up on photosystem 1. So now they're low energy, and instead of going on to oxygen as a terminal electron acceptor, instead they get transferred to P700 plus, which is right here. Okay? It takes that electron, there's light, kicks that electron to a high energy state, and now that electron can be attached to NADP plus to generate NADPH. So this here is the overall figure with all 8 steps, okay, of linear electron transport, which in the common parlance is referred to as the Z scheme of electron transport, because the electron goes low energy to high energy to low energy to high energy, and then it sort of stays high energy here. Z scheme, all right? Questions about this, please?
So these chlorophyll molecules that absorb the specific wavelengths are in the same reaction center.
The special chlorophyll molecules, so if we look at the photosystem, it has a reaction center complex and it has light harvesting complexes. The special pair of chlorophyll molecules are in the reaction center complex. Yes, and they're dimerized, so they're linked together. Yeah, they're bound together. Kind of makes sense.
Thing is happening in one space.
In one protein blob. Yeah, absolutely. It's like a little power reactor, but all it does is make high energy electrons. That's what a photosystem does. It absorbs light and it makes high energy electrons. Question?
There's some little holes in my legs. Basic physics, chemistry. When you're talking about low energy to high energy, are you literally talking about like filling like higher valence shells, or?
That's what a lot of it is. Yeah. A lot of it is just, you can think, so there's two ways you can think about it. You can either think about the electrons are starting at farther out valence shells and then falling down to lower valence shells, or you can think about it as these members of the electron transport chain increase in electronegativity, and so when the electron gets moved on to another one, it's at a lower energy state. Yeah, I'm using very general language of lower energy state to try to, avoid that confusion. Yeah. The electrons going to higher valence shells is really more important when we're talking about the pigments than when we're talking about these electron transfers. Yeah. Because the members of the electron transport chain are all confusing, complicated things. Yeah. Awesome. So here it is. This gets you the two things that you need from the light reactions, the ATP and the NADPH. You have energy in the form of ATP, and you have, this is a new term, reducing power in the form of NADPH. That's going to allow you to take a carbon dioxide molecule and make a glucose molecule, because you have reducing power, so you can throw electrons onto it, and you also have a straight up energy source here in the form of ATP. So you can do coupled reactions, right? Anyway. Here's a simpler model, okay? Photon elevates an electron in photosystem 2 to a high energy state. You let that thing roll down a ramp, spins a little wheel, little turn wheel thing. That generates ATP. Another photon over here kicks that electron up to a high energy state again. All right? And then you keep that electron at a high energy state in NADPH. So energy level would be on the y-axis if this was a figure. But does this kind of make sense? If you're thinking about energy as potential energy as position, the electron in this bucket still has energy. It could still fall on this unfortunate worker's head. All right. Thankfully, they're all wearing hard hats. All right. I like how they gave them like love handles in this illustration. It's like kind of funny, right? Like, why did you do that? You didn't have to. All right. So this is linear or non-cyclic electron flow. There's another process called cyclic electron flow. If you've understood linear electron flow, then cyclic electron flow is super mellow, because all cyclic electron flow is this. It only involves photosystem 1, and the electrons just keep kicking back through the electron transport chain to generate ATP. Kind of makes you think of fermentation, right? There's questions about if this is a survival strategy that lets the organism get rid of extra light energy, or if it is just a holdover from evolution, where evolutionarily, photosystem 1 almost certainly evolved first because it can carry out both an electron transport chain and generate NADPH, the two things you need to carry out the Calvin cycle, right? Cyclic electron flow. Cyclic electron flow. All right. Here's cyclic electron flow. It just makes ATP. It doesn't release any oxygen because that happens in photosystem too. All right? If this is a free living thing and all it needed was ATP, you can see how this process could have evolved, right? It's like, oh, whenever there's light, bless you. Whenever there's light, I have ATP. And that might be enough to keep something alive, especially in a world where there's not that much complexity to the things you're competing with. All right? Cyclic electron flow only generates ATP. It generates no oxygen. It just keeps recycling the same electron. Cool? Sick! There it is again. Questions? Okay. Now, this is step one, this is step two. I'm just going to show you the whole thing. Okay? I want to show you the whole thing at once. This is the whole picture. Here's the thylakoid membrane. Here's the thylakoid space. This out here is the stroma. All right, that the thylakoid membrane's floating in. Okay? Light hits, high energy electron transport chain, pumps protons. At the end of the electron transport chain, the electron gets kicked up to a high energy state again. Bloop! It goes over here to NADPH. You got this proton motive force. ATP synthase harvests it. This is just respiration again. You generate ATP. But now, NADPH and ATP can both go into the Calvin cycle. Because you'll notice, at this point, you haven't made any sugar. And you know that that final output of photosynthesis should be sugar, right? So you generate all this ATP to make glucose so that you can break it down later to make ATP. It's kind of ridiculous, except that from the beginning, I've been laying out for you, right? These molecules all have different stabilities. You can't only have ATP when it's light out. That doesn't work for you, all right? What if it's cloudy for a week? You need some sugars because they're stable. You can store them, break them down when you need them. You can continue doing stuff at night. You see what I mean? All right, so that's why the plant doesn't just make ATP with cyclic electron flow and then call it good. Solid? Great. That's the light reactions. Okay? That's it. We can come back if anybody decides that they would like more clarification on anything there. But now we're going to move into the Calvin cycle. Okay? Calvin cycle. It involves a super important enzyme, the most abundant enzyme on the planet, and an enzyme that, due to its structure, limits agricultural production by as much as 30 to, in some cases, 70%. All right? Maybe can you tell I've done that spiel a few times? So I'm going to talk to you about that. We're going to talk about different types of photosynthesis that all have to do with this stuff. Anyway, what is the Calvin cycle? The Calvin cycle uses carbon from carbon dioxide, electrons from NADPH, and energy from ATP. Then it makes this molecule called G3P. And then that G3P molecule is basically a foundation molecule that's used to make a ton of other organic molecules, including glucose. So I'm not going to test you on G3P, right? We'll talk about photosynthesis as generating glucose, even though you know that it's never that simple. All right? Great. So here's the Calvin cycle. Disgustingly oversimplified. As a plant physiologist myself, having to memorize the Calvin cycle in its entirety, I look at this and it makes me want to fucking puke. All right? going to be nice and simple, and I'm not going to ask you to memorize it. This is not a class where I ask you to memorize cycles, okay? I want you to know what goes in. I want you to know in general what happens during the cycle, and I want you to know what comes out. Cool? Solid? All right. Calvin cycle. The input is carbon dioxide. There is an enzyme called rubisco, which is short for ribulose bisphosphate coenzyme, I think. Oh no, ribulose bisphosphate carboxygenase oxygenase. It's not a coenzyme because it can both carboxylate and oxygenate rubisco. Ribulose bisphosphate, I mean. I'll explain it in a moment. Yes?
Is Rubisco that most abundant enzyme you were talking about?
It is. This is, and also points. You can have 2 for that one. It's very good. Rubisco is incredibly important. It's the most abundant enzyme on the planet by weight, based on, estimates. Fine, right? It's crappy. It does its job poorly, all right? It evolved in an atmosphere where there was no oxygen, and the fact that it now has to deal with oxygen, it doesn't know what to do with it. It keeps screwing up by taking oxygen into this cycle instead of CO2. It's called photorespiration, and it is not positive in any way. It's an entirely negative process. It's not even like fermentation. You make poison, but you get something out of it. You get some ATP. Photorespiration, you just ruin ribulose bisphosphate. That's all you do. So it's bad, all right? Now, why haven't they fixed it? Why can't a plant biologist fix it? They're trying. Boy, oh boy, are they ever trying. Why hasn't it evolved to be different? The plants have found ways around it. Yep. But evolution is, as an engineer, an opportunist, not a perfectionist. And if something works, it'll usually persist. Especially if you have to change something as complicated as the full structure of an enzyme, right? Question?
This is what you're describing as being like limiting agricultural growth, just like literally limit like vegetation growth on the planet?
Yeah, the idea is if the plant was not carrying out photorespiration, it would be 30 to 50 to 70% more productive. Yeah, and photorespiration only occurs because of this dumb enzyme right here. All right? Yeah. So, let's talk about this. There are 3 main stages that your textbook focuses on: carbon fixation, reduction, and release of G3P from the process. All right? Here's what I want you to see. This cycle consumes ATP, all right, and NADPH. So it's consuming that energy in order to generate this G3P molecule, which is a 3-carbon molecule with a phosphate group on it. That's where the 3 and the P come from. And then this molecule can be used to make glucose and other compounds. You started with carbon dioxide and you end up with this. This is a lot better. It's got more carbon, it's got more energy. It's even got a dang phosphate group sitting there, which is like an energy source, right? You can snap that phosphate group off and release a bunch of energy. It's so reactive. Cool? All right. All right. Carbon fixation, CO2 gets attached to this molecule, this 5-carbon molecule called ribulose bisphosphate. Okay? That makes a 6-carbon molecule, which then immediately breaks down into 2 3-carbon molecules. Look at all the phosphate groups all over the place here. That's from all the ATP that's getting consumed. It's just providing energy for these complicated processes. There's a ton of enzymes going on. It doesn't make a lot of sense because it's oversimplified. So you can't think about it too much. You know what I mean? Yeah. So our key thing is carbon fixation and then producing the glucose. But I show the figure to be like, here's how the textbook explains where all the ATP and NADPH goes. Does this make a little bit of sense? Cool. Cool. Here, let's just... Sometimes I feel like I need to justify myself. Here we go. Oh, they never give me a good image. Well, this is better. So while it's kind of a cyclical process, it's also not really. The molecules kind of like zip around between these things, right? It's a whole mess. Okay, and this is even an oversimplified version of the Calvin cycle. All right, so this is why I tell you to not worry about memorizing this, because it doesn't accurately represent the Calvin cycle. Question, please.
Will you talk a little more about the glucose coming off? It says it makes G3P.
Yeah.
There's 2 arrows and it says glucose in the compound.
Totally. I'm gonna be lazy. All right, G3P pathways diagram. So G3P can be used to make a bunch of different things by entering into different enzyme catalyzed pathways. Okay, I thought this would be easy to find a nice simple one. No, it's not very great here. It's not really great here. How about this one? Glucose breakdown, G3P, pyruvate, lactate, boom. Here you go. Okay, cool. So you've got your G3P right here. It's a 3-carbon compound, just like glycerol. It can be really easily converted into glycerol, okay? It can move into all these other different types of molecules, which are catalyzed. These reactions are all catalyzed by specific enzymes. You can see right here, multi-step pathways. This one generates pyruvate, which you might remember from earlier as a breakdown product of glucose. And that pyruvate can be used to drive. It can actually be pushed directly into respiration as pyruvate without needing glycolysis. So some of your G3P just makes pyruvate that then can be more rapidly used. Some is used for making glucose, all right? You can drive these pathways in opposite directions by adding ATP in, okay? I mean, an additional step, a pretty easy additional step, can take pyruvate and turn it into a signaling molecule that causes cells to do different things. The idea here is that G3P is a very general purpose building block 3-carbon molecule that's holding a phosphate group. So G3P, the great thing about it is it's reactive, meaning that when it goes in, when an enzyme picks it up and then picks up the other thing that it's going to react it with, breaking that phosphate group off already provides the energy you needed to make the larger molecule. Does that make sense? That's why G3P is such a valuable output. But discussing G3P in any more detail, you would want to talk to an actual biochemist. And I'm just I'm a barely passable physiologist, which a physiologist is a biochemist who hates biochemistry. It's an oversimplification as well. Question? I see a question? Yes.
I was kind of curious about like a big picture, like wondering how forms of energy are different between an ADPH and ATP. Like an ADPH makes sense that its energy is released in the like transfer of electrons. Is that the same as ATP? Or just like
No, it is different. The idea of a bond breaking and energy being used is frustrating. You can think about it more as, if you want to make it jive more, what's the word I'm looking for? If you want to make it sync up better with the concept of electron transfers, you can think about that phosphate group as an energy source itself, right? So if I'm ATP, I've got 3 of these phosphate groups coming off of me. And oftentimes what'll happen is, I wonder how